This is part 2 of 2 on chapter 26, looking at environmental microbiology. So in the first part, I kind of just went over what ecology is, the different types of ecology, so how it's divided up into, you have the biosphere, and then you have biomes and ecosystems, communities, populations, and then individual organisms. And we looked at the energy and nutritional flow through these ecosystems, and then also a little bit on cycling, so the atmospheric and sedimentary cycles. This part of the chapter is focusing more on the lithosphere, so we're going to look at soil a little bit, and also the hydrosphere. So first off, we're going to look at the lithosphere, which is the soil microbiology. So soil doesn't seem too exciting, but it's actually a very dynamic ecosystem that has lots of interactions in it. So we have the geologic part, so if you have sand or clay, or what types of rocks you have in there. There's chemicals, so chemical cycling happens in the soil. We looked at, there's phosphorus cycle that happens, it's a sedimentary cycle. And then you also have the biological factors, so we have lots of different fungi and insects and bacteria that live in the soil, and also plants grow in the soil. So if you look on the right, you can see that there's actually a lot going on in the soil. So we have the big sand particles, and then we can also have mites, so we have arachnids, so we have the roots of plants growing in there. We have Mycorrhizae hyphae, which is a fungal. We have another type of fungus, so that's the orange and the purple string-like looking things up at the top. You also can have bacteria in there. You have protists, different types of protists, which are the animal-like organisms. You can also have nematodes, which are the roundworms in there. So you have a lot of stuff going on in the soil. So like I was saying, this is looking at the lithosphere, which is looking at the soil. And there's different types of the lithosphere. So the humus or the humus, this is the rich, moist layer of soil that contains plant and animal debris, so it's very high in carbon. And it also has lots of microbes in it that are decomposing this debris, so it includes waste, or it includes dead plants and animals. There's the rhizosphere. This is the zone of soil around plant roots that contains associated bacteria, fungi, and protists. So Mycorrhizae, for example, is an association between a fungus and a plant. The root nodules that we looked at in the previous section are a symbiotic relationship between a plant and a bacteria. So that's the rhizosphere. Here's Mycorrhizae down at the bottom. So that's a symbiotic relationship between a fungi and certain plant roots. So again, here's a symbiotic relationship. This is a mutualistic one. So that was very quickly soil microbiology. So this chapter is really short. I'm not going to go into a lot of detail about it, but we have to cover it. And it goes well with this unit. So we have the final part is our hydrosphere, or the aquatic microbiology. So water, we know that it makes up about 75% of the Earth, so it's a very dominant compound on the Earth. It occupies 3/4 of the Earth's surface. And water is continuously cycled as well, along with the carbon and the phosphorus and the nitrogen that we just looked at in the previous part. So water also goes through a cycle, and it's cycled between the hydrosphere, so the water part, the atmosphere, so the air, and the lithosphere, or the soil. And this is called the hydrologic cycle, or the water cycle. So again, these cycles, I'm not going to go into a lot of detail, but the water cycle, we'll start off with the atmosphere. So you have water in the atmosphere, and if it forms clouds, you can have the precipitation in the form of rain or snow or sleet or any of those types. So this water, it goes from the atmosphere, precipitates out, it can land on the earth over here. So the lithosphere on the left, we have precipitation landing on that. It can land in the hydrosphere, so which is on the right side. We have water coming down, landing in water, or lakes, or oceans, or rivers, or any of that. This water, it can be in the ground, so we have groundwater. We can have aquifers that are even down deeper as well. So water goes from the air, goes onto the land or into water features, and then it will evaporate back up into the atmosphere up at the top. So again, if you kind of look, you have the cycle where it's going up and down from air to the land and the water. So in aquatic ecosystems, if we look at these water features, so if we specifically look at lakes, lakes are stratified vertically and horizontally into several zones or strata. So if we look at the vertical zones, which is going up and down, so you can see there's the Photic Zone, and this is on the right-hand side. The Photic Zone, this is a zone that light reaches. So you have a lot of light at the top, and then eventually the light can no longer go any further. So the Photic Zone is any as deep as the sunlight can go. Below that, once the sunlight can go no further, you have the Profundal Zone, and this is in the dark. So those are the vertical zones. The horizontal zones, we have, there we have a compensation level. That's the level where the light can no longer go down. Alright. Here. So we talked about the vertical zones. The horizontal zones, if you look at the picture down here, horizontal zones, you have the littoral zone. This also goes with the compensation zone, so it's as far into the body of water that you'll have light. And then the limnic zone is the water part after you reach that limit, and the benthic zone is on the bottom, so the soil part of your lake down here. In the photic zone, so the zone where light can reach, you have your phytoplankton that can do photosynthesis. So we have aquatic algae, we have blue-green bacteria or cyanobacteria, and we have diatoms, which also can do photosynthesis. And then you have the zooplankton. Those are animal-like organisms that eat the phytoplankton. So these things rely on having that sunlight available up here. In the benthic zone, you'll find things like bacteria that are heterotrophs, so they don't need sunlight. They can eat other organisms. You can also have larger organisms like a mollusk. You can have worms or crustaceans that live on the benthic zone at the very bottom. So besides aquatic environments or freshwater, we also have marine environments in the hydrosphere. So marine environments, they resemble that profile of the lake we just looked at, except they're a lot bigger. And you also have things called an estuary. This is where the freshwater or a river meets the sea, and it fluctuates in salinity or salt level, and it's very high in nutrients. So, and then in addition to the zones that we just looked at, marine environments have an abyssal zone. This is extremely dark, extends to depths of 10,000 meters, so very high pressure, very dark, and it supports communities of organisms with very extreme adaptations. So they have to adapt to very high pressure. So if you remember, we talked about barophiles. This is where you'd find those organisms. Right, so these aquatic communities in the photic zone, it's the most productive, and I kind of mentioned this already. It contains the plankton, the phytoplankton, which do photosynthesis, and the zooplankton that eat those photosynthetic organisms. The benthic zone supports a variety of organisms, including aerobic and anaerobic bacteria, and some decomposers as well. So the benthic zone, remember, light doesn't reach the benthic zone. It's at the very bottom of the lake or the ocean. So these organisms have to be heterotrophs, or they have to be anaerobic bacteria. They do not have oxygen available to them. So lake profiles, large bodies of standing water develop what's called thermal stratification. So there's the upper level, which is the warmest level, epileminon. They're very hard to pronounce, sorry. There's the hypolimenon, which is deeper and cooler. So that's the very bottom of the lake. And then in between them, there's the thermocline. This is the buffer zone between the warm and cool layers. And usually it prevents the mixing of the two. However, the two layers can mix with each other during certain times of the year. So the cycling that I talked about where the two layers actually switch, so the bottom goes to the top and the top goes to the bottom, this happens during the spring and the fall, and it's brought on by temperature changes. So you get this upwelling of nutrient-rich benthic sediment, so the nutrient-rich bottom sediment, and it usually causes outbreaks of microbial growth, like algae, or you can even get something called red tides, which is toxic to organisms and humans as well. So in the fall and the spring you get, well, I'm from Minnesota, so I kind of know more about this, but it's called seasonal upwelling, or you get the rotation of the lakes, and it usually causes the water to taste really bad because you have this algae bloom and you can smell it. So in those two, that's where the two layers will switch with each other. So these red tides, so if this switching causes a red tide, this is a type of dinoflagellum that produces a neurotoxin, and this neurotoxin can cause fish kills, or it can cause problems to humans if they go swimming in waters that have the red tide in them. One important part about water is looking for drinking water, or potable water. So this drinkable water, it's free of pathogens, it's free of toxins, turbidity, odor, color, and taste, so it can be used for drinking. So the most prominent waterborne pathogens include Giardia, which causes diarrhea, Cryptosporidium, Campylobacter, Salmonella, which also causes diarrhea, Shigella, Vibrio, Mycobacterium, HAV, which is a virus, or NOAC viruses. So most of these pathogens just cause diarrhea and very uncomfortable. So in microbiology, some microbiology labs are responsible for testing drinking water to make sure it's okay. Most of the assays or testings that are done of water purity focus on detecting fecal contamination. So specifically they look for E. coli, enterobacteria, or citrobacteria. And these bacteria also cause diarrhea. So we're going to look at some of these methods to screen for drinkable water. So water quality assay, there's the standard plate count, which we talked about. So this is where you take your sample, you can dilute it down a few times, and then you just plate it and put it in the incubator and then take it out and count the number of colonies that develop. And this helps you kind of estimate the total population in your sample. There's also another method called the most probable number. It's a little more specific. It's a presumptive test, so you can't use it to say yes, there is E. coli in there. It's just presumptive. You have to do more testing after the most probable number test, and I'm not going to go into detail of that one. Another assay is the membrane filter method, which is a really good method, so we'll look at them more closely. So this membrane filter method, you take your water sample and you filter it through a membrane that has very, very tiny little holes in it. So these holes are small enough to let the water get through, but they do not let any bacteria get through. So very, very tiny, very cool, small membrane. So you filter the water through this membrane, and then you take this membrane and you put it on an agar plate, and then you incubate it. So this agar plate, it can, you'll put it under a black light if it fluoresces, so I'm up at the top on the right. If you have colonies that fluoresce, these are coliforms. This means that these bacteria are ones that you find in fecal matter. And then if you look at under natural light, E. coli colonies will appear blue under natural light. So you can spot E. coli colonies, which this one's positive for E. coli in the middle. And then this is what it looks like under normal light at the bottom. So you can see there's a lot of colonies. Some of them are blue, some of them are going to fluoresce under the black light up here. So this is a really good method to actually count the number of colonies, so it's kind of like a standard plate count, and then you can actually tell if some of those colonies are E. coli, so you can actually identify them and say, yes, there is E. coli in the water sample that we filtered.